and my group members had come out with creative learning tools for the student. We have chosen an interesting topic on a muscle. We are going to explain on the structure of a muscle, the process involved in neuromuscular junction, and the muscle contraction cycle. We have done a project on how a student can understand carefully and well on a task and how it will be suitable for him. All that happening by applying many methods. We hope so it can bring more benefits in student life. Let's roll.
And so a skeletal muscle cell, as I mentioned in the introduction video, is cylindrical shaped and multinucleated. And a skeletal muscle cell is known as a muscle fiber. And interestingly enough, skeletal muscle cell, the fibers, are striated by a highly organized internal arrangement. So this muscle fiber has an internal arrangement. So if we pull pull some things out from this muscle fiber, we can find myofibrils. So muscle fiber contains many myofibrils. And if we take a portion of this myofibril, we can see that it's made out of some interesting structures known as the thin and thick filaments. So the myofibrils contain thick and thin filaments. The red is the thick and the blue is the thin filaments. But of course, the muscle fiber, the muscle cell, is actually part of a bigger organized structure. So if we look at this bone and a skeletal muscle which attaches on it, and we cut a cross-section of skeletal muscle, we have the muscle, the organ. And so this is what the muscle looks like as an organ. And the muscle contains, if we look at the cross-section, different portions of the muscles, and we'll look into this soon. But the muscle contains an outer connective tissue layer known as the epimysium, and I hope I pronounced that right. And so we take one of these portions of the muscle here. The portion of the muscle is known as a fascicle, and the fascicle also contains other internal structures. And the outer sort of connective membrane tissue is known as a perimysium. And then if we pull out one of these other internal structures of this fascicle, the portion of the muscle, this is where our muscle fiber is. And the muscle fiber contains a connective tissue wrapping around it known as the endomysium. And remember that a muscle fiber is the muscle cell. So this is this. And also the muscle fiber, the muscle cell, contains a membrane known as a sarcolemma. And again, if we remember, if we take one of the internal structures of the muscle fiber, we have a myofibril. And the myofibril contains the thin filaments and the thick filaments. So let's just recap. If we start off with a whole muscle, an organ, and we get a portion out of that, we get the muscle fascicle, which is a portion of the muscle. And the muscle fascicle contains many muscle fibers, the actual muscle cells. And then again, the muscle fiber, the muscle cell, contains many internal structures, the myofibrils. So muscle fiber contains many myofibrils, which are specialized intracellular structures. And the myofibrils are the ones that contain thick and thin filaments, also known as the cytoskeletal elements. And so we will concentrate on the myofibrils, the specialized intracellular structures of the muscle fiber. And we'll specifically look at a portion of the myofibril. And the portion of the myofibril, they all contain the thin filament here in blue and the thick filament here in red. And an important terminology to know is a sarcomere, and a sarcomere runs from one Z line to another Z line. So from basically one thin filament Z line to another thin filament Z line. And so if we zoom into this one sarcomere, here we have the thin filaments made up of actin, and the center of the sarcomere we have what's called the M line. And from the M line in the center we have the thick filaments, which are made up of a protein myosin. And we have other internal structures here in orange, which we will look into later on. But remember, sarcomere runs from one Z disc or Z line to another Z disc to another Z line. And this is the thin and thick filament structure, a sarcomere structure. But let's have a closer look at one sarcomere. So here we have one sarcomere, with the center being called the M line here. And the sarcomere runs from one Z disc to another Z disc, or a Z line to another Z line here. And a sarcomere is a section and extends from one Z disc to the next Z disc. And the sarcomere is the smallest contractile unit of muscle, so you can say. And so when a sarcomere contracts, or when a lot of sarcomere contracts, this is when our muscles contract, when we move, for example. And so again, looking over the anatomy of the sarcomere, we have here the thin filaments, which are made up of a protein called actin. And then we have the thick filaments, made up of a protein called myosin. And then these orange things, which connect to the thick and thin filaments, are elastic filaments, made up of a protein called titin. But the most important thing to know from this structure is that myosin forms thick filaments and that actin protein forms thin filaments.
This is the figure of the neuromuscular junction. An axial potential arrives at the presynaptic terminal and causes the voltage-gated calcium channel in the presynaptic membrane to open.
Calcium ions enter the presynaptic terminal and initiate the release of the neurotransmitter acetylcholine from synaptic vesicles.
Acetylcholine is released into the synaptic cleft by exocytosis.
Acetylcholine diffuses across the synaptic cleft and binds to ligand-gated sodium channel on the postsynaptic membrane.
Ligand-gated Na+ channels open and Na+ enters the postsynaptic cells, causing the postsynaptic membrane to depolarize. If depolarization passes threshold, an action potential is generated along the postsynaptic membrane.
Acetylcholine unbinds from the ligand-gated sodium channels, which then close. The enzyme acetylcholinesterase, which is attached to the postsynaptic membrane, removes acetylcholine from the synaptic cleft by breaking it down into acetic acid and choline.
Choline is imported with sodium into the presynaptic terminal, where it can be recycled to make acetylcholine. Acetic acid diffuses away from the synaptic cleft.
Acetylcholine is reformed within the presynaptic terminal using acetic acid, generated from metabolism and from choline recycled from the synaptic cleft. Acetylcholine is then taken up by synaptic vesicles.
Hi, today we are going to talk about contraction cycle. There are a few steps in contraction cycle. First, exposure of active sites. Before cross-bridges cycle, calcium binds to the troponins and the tropomyosin move, causing the exposure of active sites on actin myofilaments.
So the second part is cross-bridge stage. Myosin head bind to actin, form a cross-bridge. So as you can see, the head of myosin will bind to the exposed actin to form a cross-bridge, and the phosphate will release from the myosin head. Alright, now we are going to the next stage. This is a power stroke stage.
myosin cross bridge rotate toward the center of sarcomere. If you can see, energy stored in the head of myosin is used to move the head of myosin, causing the actin of myofilament to slide past the myosin myofilament, and this ADP is released from the head of myofilament. Next, I will talk about the detachment of cross bridge. As myosin heads binds the ATP, the cross bridges detach from the actin. As you'll see in this figure, an ATP molecule, it binds to each of the myosin heads, causing the cross bridge to detach from the actin.
Okay, now I will talk about ATP hydrolysis. Myosin head hydrolyze ATP and become oriented and energized. Myosin ATPase portion at the myosin head will split the ATP to ADP and phosphate, so it will remain attached to the myosin head.
Now, let's look at the last stage in the contraction cycle, which is the recovery stroke. In this stage, the heads of the myosin molecules return to their resting position and energy is stored in the heads of the myosin. If calcium are still attached to troponins, cross-bridge formation and movement are repeated. At this point, let's take a little recap on the previous stages in contraction cycle. The first stage is cross-bridge formation. The second stage is the power stroke. The third stage is the detachment. The fourth stage is the ATP hydrolysis. And the last stage is of course the recovery stroke. Now, as a conclusion, this cycle occurs many times during a muscle contraction, and not all cross bridges form and will be simultaneously. I hope this video has helped you all in understanding a little about this contraction cycle. Thank you, until we meet again, bye-bye.